In this video, we will continue our discussion about this FCPR03 that blocks neural damage by reducing the ER stress. In the previous video, we have uh, discussed some of the experiment that support this point, that is FCPR03 works by reducing the ER stress, and it works similar to that PDE4 inhibition works. So here is the diagram for how the PDE4 inhibition works, and in this figure we will discuss one of the additional point that supports this fact, or that supports this argument that FCPR03 works by reducing the ER stress. So let us quickly revise how this PDE4 inhibition works. The PDE4 inhibition, the PDE4 inhibition, the CAMS formation will be move towards this NRF2 formation. CAMS will move towards, helps in the NRF2 to move into the nucleus, so that this NRF2 will activate more formation of this hemooxygenase 1, and that will neutralize this reactive oxygen species. And we know from the literature that more the reactive oxygen species, it will cause more ER stress. So how this PDE4 inhibition works? PDE4 inhibition will leads to more amount of this NRF2 go into the nucleus. Then it will trigger, or we can say it will trigger a transcription factor in a way that it will increase the production of hemooxygenase 1, and that will neutralize ROS. Thus the ER stress will be reduced. So in this panel, what the scientists have, or the researchers have done the experiment, is that they inhibit this hemooxygenase 1 in the FCPR03 treated cells, so that this ROS will not be neutralized and we will be able to see the apoptosis. So this is another way, this is, we can say, another approach to check whether FCPR03 works by reducing the ER stress or not. So this is the Western blot of HO01, and this is done using different condition. This SIHO1 is the silencing RNA that is been transfected in the cells. So in first case it is not transfected, only the ER stress is produced through this oxygen glucose deprivation. And in the second case we can see that cells are treated with the FCPR03, and then oxygen glucose deprivation, ER stress is also produced, and we can see that the increase in the amount of HO01 as cells are pre-treated with FCPR01. In the next, what has been done is that cells are treated with this silencing HO1, or transfected with this silencing HO1, and we can see that they are decreasing the amount of HO1 production. And there is, even though, and also not even though, and also this this cell in this condition is been under cells in this condition are under ER stress through this oxygen glucose deprivation condition. And in the third case we can see that in all the three conditions are positive, that is, cells are first transfected with SiHO1, so that this HO1 production is reduced, and then pre-treated with FCPR03 for 1 hour, and then kept under ER stress or oxygen glucose deprivation condition for 6 hours. And that the Western blot is done of the cells, and we can see that there is a decrease in the amount of HO1 production compared to the second case. And same thing they have done to check whether the proteins that are involved in the ER stress get upregulated or downregulated. And we can see that in the third case there is upregulation of the proteins. In the last case we can see there is upregulation of the protein compared to the case where there is no silencing HO1. So this indicates that the HO1 could be the reason, or FCPR03 might be somehow triggering this NRF2 to go into the nucleus, and that will form HO1. And when we inhibit this production of HO1, we can see that the ER stress got increased even when the cells are treated with FCPR03. When they check with other proteins, that is PEIF2 alpha, we can see that it is also increased. And the panel BDF is the semi-quantitative analysis of the Western blot to confirm whether there is an increase or not. And we can see that uh, in the case when there is no silence HO, the production of HO1 is more, and compared to the case when the cells are pre-treated with FCPR01 and cells before that cells are treated with SiHO1 and then with oxygen glucose deprivation, we can see that there is not much increase in the HO1 production. And this is because we have added the silencing HO1. So this in a way prove the point that this silencing HO1 is functioning properly. And in the another case where the proteins involved in the ER stress are measured, we can see that compared to the case where there is no silencing HO1, the production of GRP 78 is low, or the ER stress is low, as HO might be neutralizing reactive oxygen species. And whereas in the case where silencing HO1 is there, the production of GRP 78 is more. And when similar is the case they have observed with phosphorylated EIF2 alpha. Next, what they did is they checked the amount of ROS that is present within the cells to see how much changes in the ROS observed in the case where silencing HO1 is there and silencing HO1 not there. When silencing HO1 is not there, the amount of ROS is reduced, as ROS might be neutralizing with this HO1. And when the silencing HO1 is when cells are treated with silencing HO1, in that case there is an increase in the amount of this proteins that are involved, increase in the amount of this ROS. This shows that ROS could be the reason that is causing ER stress, and HO is not able to neutralize this ROS the same way PDE4 function, same way in which the PDE4 functions. And the cell viability test is also been carried out, in which they can see that cells viability compare in aligning with the other experiments or in aligning with the other results of these panels. We can see that ER stress is low in the second case, as the amount of proteins that are involved in ER stress is low, and also the ROS amount is low, so cell viability is more. And compared to the case where cells are pre-treated with silencing HO1 or pre-transfected cells are transfected with silencing HO1, we can see the cell viability is reduced. And finally, they also carried out to check whether this cleaved Cas3 is produced or not, as this is the direct indicator that cells are diverted towards this apoptosis, or signal towards program towards this apoptosis. So they have checked the the cleaved Cas3 amount in different condition, and we can see that in the second case where there is no silencing HO1, the cleaved Cas3 amount is low compared to the third case where where silencing cells are transfected with silencing HO1, there is an increase in the amount of cleaved Cas3. So cells are undergone apoptosis in the third case compared to the second case. And they have quantified this thing through semi-quantitative Western blot analysis. They have quantified this thing by semi-quantitative analysis of the Western blot, and this also shows the same point, that there is a significant increase in the amount of cleaved Cas3 compared to the cleaved Cas3 in the case where cells are pre-treated with pre-transfected cells are transfected with silencing HO1. compared to the case where cells are not transfected with silencing H1. So this further substantiates the point that FCPR03 function the similar way the PDE4 inhibition function to reduce the ER stress and block and thereby block neural damage within the patient. So that's all for this video. If you need to understand this thing in detail, then you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.